Okay, hello everyone. My name is David Requena. I'm gonna tell you about my research at Rockefeller University. How can I? Maybe here. Okay, first, what is fibrolamellar carcinoma? Well, it's a very rare liver cancer that happens in one in five people in the U.S. and we don't know exactly what is the incidence in the rest of the world because it's very hard to diagnose. It is thought to be a subtype of hepatocellular carcinoma because this happens in the liver as well, and it has some of the characteristic scars that I will show you in a moment that are seen also in these cancers. But the thing is that this specific subtype happens in adolescents and young adults. I mean, in people without history of like cirrhosis or without viral infections. And it has no specific symptoms. They are very vague, what complicates the diagnosis. So by the time of detection, it has usually already metastasized because it is not usual to do like cancer screening in underage people. So this is how fibrolamellar carcinoma looks like. As you can see, it has some fiber bands that I mark in blue that are what give the name to this cancer. So what is the treatment? Well, there is no treatment. So if you detect this cancer early, you can get like surgery, partial resection of the liver or the whole liver transplantation, but just 50% of the people that undergo that procedure reach 5 years survival. And there is no alternative treatment approved. And for people that uh, get this cancer detected too late, there is like just 1 year left. So that's the scenario. Um, so now I'm going to tell you about the genomics of FLC because when I started working in this lab, this was like we were kind of lost. There was no clue about what causes this cancer. Because it has a very stable genomic landscape with low mutational burden. There were no recurrent chromosomal alterations, translocations, duplications. I mean, nothing where to start. So then we checked the transcriptome and we found one single recurrent event that is just constant at transcript level, but not at genomic level. So somehow the deletion happens to like join uh, to join the exon 1 of DNA JB1 with this gene PRKK that happens to be the active subunit of protein kinase A, that is the main kinase of the cell. So as you know from basic biology, if we are changing the first part of the protein is going to go somewhere else. And this is a kinase that is going to phosphorylate things in other regions that it shouldn't. And this fusion protein that we discover, and you actually discover a new oncogene, retains the kinase activity, so it's functional. So then we have a constant mutation, we have a point where to start, but then we ask what triggers fibrolamellar carcinoma? Is this fusion? So what we did is we removed the genes in between these two genes connected that we found. So we developed a CRISPR-Cas model when we reproduce a deletion, and we develop another model using the Sleeping Beauty transposon, where we just provided the fusion protein, and in both cases we got a similar histological phenotype that looks very like the original tumor, and it is also similar at transcriptome level. So we were advancing into the study of this cancer, and then other tumors that happen also in the liver or the pancreas were found with similar histology, but missing the fusion that we found. They were even more rare. And these other tumors had other mutations that also affected the protein kinase A catalytic subunit. So the obvious question was like, are they also fibrolamellar carcinoma, or they were a different disease? So as we found the clue at transcriptome level, we went to study what happens with these other mutations in the transcriptome. We checked the previous studies, but the problem was that there were small, they had different kind of problems, like small data sets, a high number of unpaired samples, or they use surrogate samples from other parts of the body, or some benign tumors. And people tried to integrate these studies, but they were done by different groups in different times. So they did not even have the same extraction method, not even to mention the sequencing method. So it was not possible to reconcile them. The other problem is the diagnosis. It was very hard, as I mentioned. So even if you ask physicians to diagnose just based on the histology, their diagnosis didn't match. So it was crazy. Like, there was a whole mess in the literature, and one of these studies also had like, well, some had this problem of using algorithms that have like problems with quantifying reads. So what we did was we addressed all of these points, because when we compared these studies, they had very poor agreement. Like, they just agreed in 16 to 40% at most in the differential expressed genes that each one reported. So we address all of these points with a larger data set, with more per samples, a proper integrative analysis, a reliable criterion to select fibrolamellar, because now we use a dual criteria. We use the histology plus, we use the consensus histology of pathologies plus having the fusion that we detected. So we just started studying the cases that we trust that are fibrolamellar carcinoma, and with this starting point, we extrapolated to others. So we ended compiling the largest collection of liver cancers, and especially fibrolamellar carcinoma. We released 220 fibrolamellar carcinoma samples, and for a very rare cancer, it's very unusual, and about 100 per tumor normals. And we characterize those in different omic levels, with the start of a state-of-the-art multiomic methods. So I'm going to show you what we found. So the strategy was like, we had three big libraries that we analyzed. They had similar processing methods. So we got the differential expressed genes, used the intersection, and we call these three libraries our exploration dataset. Then we used two smaller libraries for refinement. And then we did use the studies made from other groups as a validation. So we did not combine them, but we validated with each one of them. And we found that the genes that we obtained through this methodology agreed in 100% of the cases with the trend that we found in the external datasets. So we ended getting some genes that always have the same trend, independent of who processed that or how this lab processed that. So with this gene set, we started doing some analysis. So I will show you, well, this is how the samples from different groups look when you plot them in a UMAP, and this is what happens if we only use the fibrolamellar transcriptomic signature that we found. We can see at the top that all the FLC tumors cluster independently of the processing method. And the normal samples are in the opposite corner, and we were able to distinguish the FLC-like tumors, and we identified that two of them were behaving as fibrolamellar carcinoma at transcription level, and one of them not. And we found later that these mutant BAP1 tumors had other mutations in other oncogenes that are probably making them like be not in the cluster of FLC tumors. So with this, we were able to identify transcriptomic signature independent of experimental processing, and we showed that what characterizes fibrolamellar is not the fusion that we found at the start, but is the activation of PKA. So we then compared this with other liver tumors because we had a big list of 700 genes. So we identified what is exclusive of fibrolamellar carcinoma, and we were able to distinguish that very strongly from other liver tumors. Okay. But we all know that when we sequence bulk tumors, there are different cell types in the tumor. So we wanted to identify how these different cells contribute to the signature that we found. So we did spatial transcriptomics, and as you can see there what is a stroma, what is a normal, and what is a tumor. So we use MERFISH, that is a technique that allows single cell spatial resolution, and we identified 3 main clusters. And when we map the top genes of each of these clusters back to the tissue, we were able to distinguish each one. So in this way, we identify how the different cell types in the bulk tumor contribute to the signature. And then we use this tool to validate like mouse models, cell lines. We showed also how under the signature, the agreement is amazingly strong. So then we wanted to connect the hyperactivation of PKA with the transcriptomic signature. So we sequenced autoromic layers. And when we did the multiomics, we found no explanation for the transcriptomic signature at genome or methylome level, but we did some correlation with the transcriptome at the proteome. So we were able to connect the dots of what is happening after, but not what is happening before. So as this is a kinase, this is maybe acting at the level of the transcription factors. So what we did is an analysis of the transcription factors that target the signature, and we found that 6 were like enriched. They preferentially target genes of the signature, normalizing by the number of targets that they usually have. And the exciting thing about this analysis is that when we try to connect the protein kinase A catalytic subunit with the transcription factors, we found intermediates, and one of the intermediates that I'm highlighting there, HDAC1, was actually the target of the drugs that we found that work in fibrolamellar carcinoma in the new screening that we did. We did an agnostic screening of 5,000 FDA-approved drugs, trying to repurpose what is already approved. And we found that the ones that work were have as target this one. They were HDAC inhibitors. So we were able to reconstruct the study. And what is the current state of this? Well, we already finished the preclinical, so we are in clinical trial phase 1. And the people that have already tried this drug, there are 7 that recovered from a coma. So this thing is working. And as concluding remarks, I already mentioned that we found what is a unifying phenotype of fibrolamellar carcinoma. So we are moving towards to defining this cancer. And we found that this is a different cancer than hepatocellular carcinoma. So we are recommending people with this cancer not to use the drugs for hepatocellular carcinoma that are usually provided in the practice because there is no approved treatment, but like try with this combination that we found. And also we found a transcriptomic signature independent of experimental processing that can distinguish fibrolamellar from other liver tumors and help to identify transcription factors that might be critical for the pathogenesis. So with that, I would like to thank all my collaborators from Sandy Simon Lab, he was my mentor, the RISE Lab that helped during this study, and our collaborators in other institutions. Thank you.
Probably you said it and I missed it. Could you please decline a bit? How did you define the transcriptomic signature?
Yeah.
Methodologically.
Yes. What we did is like we used the intersection of data sets, our biggest data sets, the intersection of the differential expressed genes. And we applied some filters that we defined in the article. Oh, but by the way, this article was just accepted in Nature Communications. So we defined some filters to ensure that these genes are detectable. I mean, that they have enough concentration, and also the consistency of the trend, at least in the libraries that we use for exploration. Then we matched this with the two external libraries that were small, so and they had different methodologies, so we didn't want to mix these two small with the other three, but we use this for refinement. We ended discarding some genes, and what was the result of that was our signature. We did not put any other definition there. Yeah. And then just the validation. We use that against the literature, and it worked.
Could you optimize that numbers because you are having there like 600 one more 700?
Yeah, when we ended comparing to other liver tumors, and when we proposed a gene set to distinguish fibrolamellar carcinoma, we ended with 35 genes. Yeah.